Afternoon. It's good to be here. I'd particularly like to thank the organizing committee for allowing me the opportunity to be here and present some research on behalf of my co-authors. I'd particularly like to recognize Dr. Stockler, who did a big part of this work, working on his PhD projects. And so just the skeleton of my talk, as you can see here, we'll talk a little bit about why this study is important and why we need to look into this sort of thing. What we did in the study, I'll show you the raw data that we got from some of this, from some of the procedures we did, and then finally discuss a little bit about what that means. And I would certainly welcome not only your questions, but any comments you may have at the end of the presentation. And so I think we know, particularly as bovine practitioners, how important the microflora in the gastrointestinal tract is to the health of our patients, particularly when we're thinking of the rumen microflora, but more and more we are learning and we're understanding that it's not just the microflora in the rumen, but throughout the GI tract that is having a big impact on how our animals are doing, not only health-wise, welfare-wise, but also production-wise. And there's lots of impacts, of course. Obviously, the first thing is metabolic functions. That nutrition is important, and that microbiome is important to getting that nutrition that they need. There's also immune system interactions between that microflora in the tract, in the intestinal tract, that will impact the immune system, and then potentially interaction and protection from other pathogens as well. Kind of the classic example being salmonellosis, potentially having some commensal flora in there that will help counteract the effects or the colonization of salmonella. Okay. What we do know is that the intestinal microbiome is really pretty poorly characterized in cattle. There's lots of studies, and certainly they're increasing in some of our other veterinary species as well as humans. But for obvious reasons, most of the studies looking at the microflora in cattle have focused on the rumen, probably for good reason. The studies that have been done further down, further distal in the GI tract, have really focused on just taking lumen samples, which may not accurately reflect what's actually going on in that epimural surface of the GI tract, or looking at fecal samples when that digested material has passed all the way through, which again may not be exactly reflective of what's happening further up in the GI tract. And so these will be some new words for some of you. Some of you are very familiar with these. Firmicutes or firmicutes, depending on how you want to pronounce it, and bacteroidetes are kind of the two main phyla that we see in GI flora. The firmicutes tend to be gram-positive organisms that are very important in energy metabolism and absorption. Bacteroidetes or bacteroidetes tend to be more of the gram-negative organisms. And these are the two main phyla that compose GI flora pretty much across species. Some of the human studies are going to say that they compose 90 to 99% of all the bacteria in the gut. And it'll be similar. I'll show you some data later. We don't get to 99, but it's certainly those are the major players that we're talking about. And what we do know is that the ratio that we see of these two phyla certainly has an impact on GI health. Not just in humans, but we're seeing that more and more in some of our other veterinary species. And so the question is, does that hold true in cattle as well? What we do know certainly is that this ratio, the firmicutes to bacteroidetes ratio, does have some implications when it comes to obesity and energy metabolism, as well as there's some characteristic changes with aging that happens in humans that you can profile going along. We're not quite there yet in cattle, but I think that is coming. So the question is, how does antibiotic use affect this GI microflora? Particularly, we've just heard a couple of really good talks that reference empirical antibiotic use by practitioners as well as farmers. And so how does that affect potentially that GI flora? At least in the United States and North America, respiratory disease is by far the most common indication for antibiotic usage. But certainly we know when we're giving an antibiotic systemically that will be absorbed systemically, absorb throughout the body and affect bacterial populations in other parts other than just the pulmonary fluid and the lungs. And so that certainly becomes important. And that's what we wanted to look at with this study, is how that systemic administration of antibiotics may affect the GI flora. And so to do that, we chose a macrolide antibiotic. We chose gomithramycin, which was one of the newer antibiotics we have available, just as a representative of the macrolide class. I probably don't need to go into a lot of this, as y'all are familiar with the macrolide antibiotics, but it is a protein synthesis inhibitor, tends to be bacteriostatic in vitro, although there's some discussion that it may actually act as bactericidal when we get to therapeutic concentrations in the tissues. The reason we like some of these macrolides is it has very rapid absorption. So particularly gomithramycin, after subcutaneous administration, we're going to reach therapeutic concentrations in that lung fluid within 45 minutes to an hour, something like that, which is pretty good. Okay? Some other benefits, very good volume of distribution, and so we can get good concentrations of antibiotics to many parts of the body where we're trying to fight that active infection, which also means we're going to get active concentrations of that antibiotic in our GI system, whether that's what we're trying to target or not. Okay. And then extended duration of action as well, obviously very beneficial for treating those animals that aren't so easy to treat, that we can't get up every day, when we can give them a single dose of antibiotic and have extended duration of therapy for those infections that need a little more treatment. Okay? And as I mentioned, the effect of this antibiotic administration on the GI tract is as of yet undetermined. And certainly there's the question, how are we affecting those GI populations either in the short term or even in the long term when we're administering systemic antibiotics to our cattle, and then potentially even further on, how does this affect the human food supply, either through dairy products or meat products from animals that may have been treated with antibiotics. Okay. So the study aim, as you can see here, is simply to evaluate the changes, if there are any, in the bacterial diversity of the enteric microbiome in cattle after the administration of a single dose of an extended duration, commercially available parenteral macrolide antibiotic. So label use, just like we would be doing on farm or in the clinic, what effect does this have on that microbiome? And so here's how we set it up. We had 6 steers, and we placed these T cannulas into the ileum surgically. Pretty standard standing flank laparotomy. You can see these T-shaped cannulas here that were purchased commercially. They are medical grade. This part here lies within the ileum. The ileum was sutured around, and then this port is actually sutured to the exterior of the body, included in the suture. So now we have a sampling port into our ileum for sample collection at a later date. Here you can see a few of the steers. You can see they are pretty happy, healthy. We did allow at least a 90-day period following surgery before we initiated therapy. As part of the surgery, they did receive a dose of antibiotic. It was not a macrolide antibiotic, it was a cephalosporin. So none of these calves had ever been exposed to a macrolide antibiotic prior to this study. Fed a consistent diet throughout the study, tried to keep things as consistent as we could, just for the sake of eliminating that source of variability within our data. Okay. So out of the 6 calves, 2 of those were controls. They were not given an antibiotic. The other 4 were given that single labeled dose of macrolide on day 0 following initial sampling. And then we also sampled at 4 other points throughout the study, ending at day 35, which coincides with the recommended withdrawal time for gomithramycin. So for sampling, we really took 2 samples from each of these calves at each sample time point. So you can see here in the right, we're just collecting a lumen sample. And so to do that, we open up the cannula, stick a sterile piece of tubing in, and suction out just part of the luminal contents that we can get in the ileum. Okay. The other sample was collected via video endoscope. So inserting the video endoscope down into the ileum, and then we can take a surgical biopsy of that ileal epimural surface to see if those populations between the lumen and the epimural surface are different or if they are the same. So all these samples were taken in triplicate at each time point, and then immediately the DNA was collected, the DNA was extracted, and then sent for deep sequencing for analysis. And so here you see just basic results. And once again, this orange color in both of these indicates the firmicutes proportion, which is about 60% in both the lumen populations as well as the epimural surface populations. And so actually very similar in this study. In some previous work that we've done in our lab, we actually saw quite a bit of variation, particularly when looking in the rumen populations and the duodenal populations. There was actually quite a bit of difference between these 2 sites. In the ileum, they seem to be pretty consistent. Second biggest population there you can see is the Bacteroidetes, and together they make up about 90% of the total reads that we got from these calves. Okay. So now for the individual calves, I'll just explain this one a little bit, and then I'll show you the other calves. This represents all the samples from one calf in the study. This was one of the control calves. So we have day 0 here on the left, all the way up to day 35, with both the epimural and the lumen surfaces showed, and you can see basically the different composition or different proportions of bacterial populations at each sampling time point. And what you see in this control calf is that it's relatively stable throughout the sampling period. Certainly there is some variability, but it tends to be pretty standard, which is what you would expect or what you would hope for, I guess, in a control calf in a controlled study. Here is our second control calf, and obviously this one didn't quite read the book. Okay, so certainly we see some changes within this calf's microbiome, even though he did not receive antibiotics. So what this illustrates is there's certainly going to be some individual variation between calves. Okay? So here's 2 of our treated calves. Just as a reminder, here's this red arrow indicates the time of treatment. So this sample was taken before treatment. And what you see, there are some changes, but we don't see a drastic or a consistent change right at the time of treatment. A little bit of variation, but then by day 35, in both of these calves, we're largely back to that pre-treatment populations. Okay? Same thing with some of these other calves. Here you do see a little bit of a drop. Now, whether this is significant, it's hard to tell. With just 6 calves, we couldn't make it statistically significant. But again, by day 35, we're largely back to normal. You may notice that this calf right here certainly has a little spike in the Bacteroidetes there at day 8. And interestingly, this calf did have some clinical diarrhea just prior to that, which I'm not sure how that's associated, but they certainly do seem to be linked. Okay. So what's this mean? We're still in the process of figuring that out, but obviously we know that there is variation in the ileal microbiome between time points on individual calves, but also between calves individually. And unlike some of the other sites higher up in the GI tract, as well as the forestomach, the luminal and epimural surface populations seem to be fairly consistent between those 2 sampling sites. We don't see the differences that were characterized or that characterize some of those earlier sites. And so what this means is that we really did not detect a distinct change consistent with or in conjunction with antibiotic administration. in the relative abundance of that microflora, at least at the phylum level. We have some of the data further on that's in the process of being analyzed, but no stark change that we saw. The changes we did see that were minor, those tended to resolve by day 35 when the study ended. And so with that, I will come to the end of my talk and appreciate your attention.